The risk of contracting cancer is increasing, and according to some recent projections, the likelihood of each of us contracting cancer over our lifetimes is rising to 50%. We need to look at this problem in a new way. We need a new way to fight this disease. One of the problems is that the conventional view of how a cancer grows is that all cells within a tumour can switch on that tumour. However, when you look closely at a tumour, you realise that not all cells are the same, and that some cells are more aggressive than others. This has led to a series of experiments where a tumour has been broken apart into single cells, and then each cell was tested to see if it could form a tumour on its own. What was found was that some cells were very good at forming tumours, but the bulk of cells were very bad. This is what the idea of a cancer stem cell is. Literally cells that are responsible for the growth of a tumour and ultimately its relapse. And the idea is that these may be used and targeted to turn tumours off.
Cancer stem cells are a very small proportion of tumour cells, which, through slow symmetric and asymmetric division, can maintain their own population by endless self-renewal, and give rise to the bulk of the tumour, by producing fast-dividing progenitor cells, which can differentiate into a variety of cancer cell types. A significant property of cancer stem cells is their ability to spread cancer to other areas of the body. They do this by breaking away from the bulk of the tumour, entering the bloodstream and circulating around the body, where they can invade other tissues and start the process of tumour formation from scratch. This deadly process is called metastasis and is a major contributor of cancer-related deaths.
Also of great significance is the resistance of cancer stem cells to conventional therapy. Current therapies tend to kill the fast-dividing cells in the bulk of the tumour, but they will also spare the slower-dividing cancer stem cells. These may survive and drive regrowth of the tumour. This is why we need to develop therapies that kill the cancer stem cells specifically. Unfortunately, even cancer stem cell specific therapy may not always be a cure, as the bulk of the tumour cells left behind may develop the ability to convert back into cancer stem cells and so drive tumour recurrence.
But if cancer stem cell targeted therapies are integrated with current cancer treatments, all of the cells within the tumour will be targeted together, destroying the bulk tumour cells before they get the chance to convert back into cancer stem cells. It is hoped this new strategy will increase the number of cases which can be cured for good.
If it's working properly, the immune system wants to kill cancer cells. That's its job. Create an army of T cells to patrol our body for signs of infection and other diseases. The T cells inspect suspicious cells and, when they find an abnormal one, immediately lead an attack on it, while limiting the damage to healthy cells. The way they do that is pretty elegant. T cells have protein receptors on their surface, and these claw-like structures latch onto antigens, protein fragments found on the surface of all cells. When a receptor latches onto an abnormal antigen, like one found only on a diseased cell, the T cell turns on and releases toxic chemicals that damage the abnormal cell and recruit other immune helpers to the area. Sometimes cancer cells develop ways to fool the immune system, either by disguising themselves as healthy cells, or by sprouting so many antigens on their surface that the T cells are stymied and can't mount an effective attack, or by finding ways to turn off the immune response against them. One innovative way to see through the deception is CAR T cell therapy. Thousands of a patient's own T cells are collected in a process similar to a blood donation, using a modified and inactive virus that can't cause any disease but can introduce genetic information into a cell. The T cells are reprogrammed so they now produce special receptors called chimeric antigen receptors, or CARs, on their surface. These newly engineered cells, called CAR T cells, are grown in a lab, and millions of them are infused back into the patient. The new receptors enable them to latch onto a specific antigen on the patient's tumor cells and destroy them.